We are examining the biochemical anchor of the human proteomic network, L-histidine. A patient finishes seared tuna. Within 30 minutes, their face flushes, hives erupt across their chest, and their blood pressure plummets into life-threatening vasoplegic shock. Simultaneously, a patient with advanced kidney disease suffers from a severe microcytic anemia that fails to respond to repeated doses of intravenous iron. These two conditions share a common origin: the physiological mismanagement of a single amino acid. Histidine is a master catalytic switch and a foundational acid-base anchor. The body treats it as high-value inventory, hoarding it in specific vaults to regulate everything from stomach acid to blood pH. Understanding these clinical outcomes requires an analysis of histidine's molecular thermodynamics, its cellular storage patterns, and the pre-analytical errors that frequently corrupt laboratory data. This 3D render isolates the defining feature, the imidazole ring. This heterocycle makes histidine indispensable to complex enzymatic networks. This gauge shows the ring's ionization constant, a pKa of 6.04. This value sits remarkably close to the physiological pH of 7.4. Because these two physiological values are so close, the energy required to either grab or release a passing proton is near zero. This allows the molecule to alternate between proton donor and acceptor states at lightning speed, driving rapid metabolic reactions without requiring massive cellular ATP. It is also a superior electron pair donor. The ring coordinates the electronic configuration of transition metals, trapping iron, zinc, and copper safely inside the active centers of enzymes. This zero-energy proton transfer capability is the thermodynamic anchor that allows human metabolism to operate within strict efficiency limits. A healthy adult holds roughly 30 to 45 grams of histidine, but very little of it circulates freely. 40% of the body's supply is sequestered inside red blood cells, bound within the hemoglobin. This is the engine of the Bohr effect. As tissues produce carbon dioxide, they generate acidic protons. The histidine residues inside the hemoglobin act as sponges, soaking up these ions to prevent dangerous shifts in blood pH. Another 35% is locked in type 2 fast-twitch muscle, as the dipeptide carnosine. This pool neutralizes lactic acid buildup during anaerobic exertion, delaying muscle fatigue. These massive vaults contrast with the microscopic fraction in the plasma, which is tightly maintained between 50 and 120 micromoles per liter. Cells regulate their own internal levels through a nutrient-sensing network called mTORC1. When internal reserves drop, this complex triggers the cell to deploy LAT1 transporters to the surface to extract histidine from the passing blood. The circulating plasma concentration is a deceptive marker. The reading reflects a transit highway dictated by the hidden demands of the deeper intracellular vaults. Improperly refrigerated dark flesh fish, such as tuna or mahi-mahi, creates a vector for massive biochemical failure. If the fish sits on a warm dock, marine bacteria proliferate and deploy their own decarboxylase enzymes. These microbes convert the fish's massive tissue histidine into preformed, stable histamine before it ever reaches the kitchen. When consumed, this exogenous histamine load completely overwhelms the body's intestinal diamine oxidase firewalls, which normally prevent dietary amines from reaching the blood. Once the firewall is breached, histamine floods the vascular bed and binds to endothelial receptors. This triggers massive vasodilation and systemic vasoplegic shock. Treatment requires aggressive fluid resuscitation and the immediate intravenous administration of dual H1 and H2 blockers. These molecules act as decoys, plugging histamine receptors throughout the vascular bed to stop the shock. Cases resisting these interventions mandate intramuscular epinephrine to force the blood vessels to constrict and restore systemic pressure. Scombroid poisoning demonstrates the cardiovascular collapse that occurs when an external source weaponizes the body's own structural building blocks. While scombroid poisoning is an external surge, a shortage of histidine creates a silent internal failure known as hypohistidinemia. In malnourished patients or those with advanced kidney disease, this deficiency manifests as a refractory microcytic hypochromic anemia. Standard diagnostic workflows often default to iron deficiency when red cells are small. But if iron stores are normal, the clinician must pivot. If they erroneously push iron instead of checking amino acids, they miss the metabolic cause. This is the iron refractory blind spot. Histidine is mandatory for building the alpha chains of the globin protein. Without it, hemoglobin assembly stops entirely, regardless of how much iron is available. Providing iron to a histidine-deficient patient is like delivering bricks to a site without mortar. The building blocks are present, but the structure cannot assemble. Liquid chromatography tandem mass spectrometry is the primary reference method for identifying these deficiencies. This instrument separates molecules by mass, quantifying the histidine based on its unique imidazole product ions. This instrumentation is highly precise, yet it is blinded by one specific pre-analytical error: mechanical hemolysis. If blood is drawn poorly or centrifuged too fast, red blood cells rupture and release their high internal concentrations of histidine into the serum. This creates pseudo-hyperhistidinemia, an artifactual elevation that perfectly hides an underlying nutritional deficiency from the pathologist. Laboratory protocols must strictly enforce the rejection of any sample with a high hemolysis index. There is no middle ground. Hemolyzed samples produce data that is actively deceptive. Furthermore, because protein-rich meals cause a transient 40% spike in circulating amino acids, a 12-hour fast is a mandatory constraint for diagnostic accuracy. Without these strict pre-analytical gates, even the most advanced spectrometry results are clinically useless. Pathologists also use histidine to detect intracellular starvation of B12 and folate through a technique called the folate trap. The final step of breaking down histidine in the liver requires active folate. If the cell is starved of functional B12 or folate, this degradation pathway hits a biochemical block. Because the pathway is blocked, the intermediate molecule Figlu accumulates, overflows the liver, and spills into the urine. Measuring this urinary Figlu after a histine challenge provides definitive evidence of functional B12 or folate starvation, long before changes appear on a blood count. Histine regulation is a core pillar of human architecture, from the 6.04 pKa thermodynamic anchor to the globin scaffolding in our marrow. This single molecule demonstrates the complexity of systems biology. A single imidazole ring directly links microscopic catalysis to macroscopic vascular integrity and survival.